All right, so we're going to look at the different stages of meiosis. Just like mitosis, what always happens before mitosis? Interphase. What always happens after mitosis? What happens right after mitosis? Cytokinesis, right? Mitosis divides up the DNA. Cytokinesis divides up the cell. So the same things happen before and after meiosis. Interphase before, cytokinesis after. What's one of the main things that happens during interphase that's important for cell division? The DNA gets copied, specifically during the S phase of interphase, replication. That happens before meiosis. That's important. DNA is copied just like it is before mitosis. So you end up with a cell, a germ cell, that after this S phase is diploid, and all its chromosomes look like that. What was the word for those X-shaped chromosomes that we've been using?
Chromosomes? Oh,
No?
chromatids.
No. Each half is a chromatid. But what's the whole thing?
Chromosome?
It's called a bivalent chromosome. Meaning that it's really what?
Two chromosomes.
It's really two. So at the end of the interphase, all your chromosomes are double like that. They're all X-shaped structures. They're all bivalent. And then we proceed into meiosis. Now remember, meiosis has those same stages: prophase, metaphase, anaphase, telophase. But they happen in two sets. So we're going to talk about the first set first, meiosis 1. The first set of PMAT. And that first step is called reductive division. Because what's it going to reduce? What's the first set of PMATs? What are they going to reduce? The number of what? Chromosomes. It's going to reduce it how? Reduce it to half. It's going to take diploids and make what? Haploids. All right, so the very first stage of meiosis, prophase number 1. Prophase Roman numeral 1. It's a lot like prophase and mitosis. Same kinds of things happen. Let's make a list. What happens to the DNA, the chromatin, during prophase? It winds up and forms chromosomes, right? Same thing happens here. The DNA winds up, chromosomes become visible. What happens to the nuclear membrane or nuclear envelope during prophase of mitosis? It disintegrates, goes away. Same thing happens here. What happens to the little spindle fibers during prophase? They form, and they form this spider web looking structure inside the cell, right? Happens here too. What about the little nucleolus, the dark spot in the nucleus? What happens to it? It goes away too. All those same things happen in prophase 1 of meiosis. But there's an extra thing that happens. Actually, two extra things that happen. And the first one we're going to call synapsis. Do you ever see the word synapse? What does that mean? If we say two people, they really synapse. What does that mean?
I've never heard that word.
It means they connect. Oh. They connect. So synapsis means connecting. And it's when the homologous chromosomes attach to each other. Let's make sure before we go any further that you know what homologous chromosomes are. What's homo mean? Same. Now, you have two of each kind of chromosome, right? But when we say each kind of chromosome, are the two chromosomes in a pair, are they actually the same? No. What's the same about them? They're kind of the same shape, same basic size. They have the same types of genes on them. Like if the chromosome you got from your dad, chromosome number 1 you got from your dad, if the first gene on that chromosome dealt with hemoglobin, what's the first gene on the same chromosome you got from your mom deal with? Hemoglobin. But it may tell you two different versions of hemoglobin to make, right? Or two different, something about the hemoglobin gene may be different. There's different forms of genes on those chromosomes. But that's what homologous means. Same kind of chromosome, same genes, same order, but they may have different versions of those genes. You have how many pairs of homologous chromosomes? In every cell, how many pairs of homologous chromosomes? 23 pairs. So the first thing that happens, those homologous chromosomes, they wrap around each other. So let's say that one's the one you got from your dad, and this one is the same chromosome that you got from your mom. They actually attach together in pairs. They wrap up. It's like they're giving each other a hug almost. While they're attached, and as they're unattaching, sometimes pieces of them break off. And the piece that was supposed to be on the red one, let me redraw that, all of a sudden ends up instead on the blue one, or vice versa. So maybe we got something like this. So the blue one ends up with a piece of the red one. And the red one ends up with the same piece of the blue one. They switched sequences of what? Genes, DNA. And what's happened is now this chromosome is different than the one you started with, right? And where's this chromosome going to end up potentially?
Yeah.
If this is happening in you, that chromosome is going to end up in your baby maybe, right? So your baby is going to get a chromosome different than even you have. This is important for creating what?
Genetic
Diversity. Making our babies different from us genetically. That's good. That's crossing over. It happens randomly. So every time this happens in you, they cross over in different places. Which means how many different kinds of sperm or eggs can you make? A crazy amount. Bigger than that number over there. Bigger than 256 trillion. Crazy amounts. Crazy amounts. So this is showing you, here's what a germ cell might start off looking like. Except ours, instead of having 4 of these, would have what? Ours would have 46, so look like that. Well, then replication happens. And replication is going to make each one of these little line-shaped things become an X. You know, like that. And then we get prophase 1. Remember, one of the important things that happens in prophase 1 is those homologous chromosomes wrap around each other. That's called synapsis. And then while they're synapsed, they switch some gene sequences, and that's called what? What's it called when they switch gene sequences? Crossing over. Here we can see them crossing over a little bit more. Switching some more gene sequences, creating new chromosomes. This is going to make your babies different from you. It's going to make them different from their mom. It's going to make them different from their brother or sister. All right, the next step is metaphase 1. And we said that meta means what? Middle. The chromosomes line up in the middle. But they don't line up single file like they did in mitosis. They line up across from who? Who is this red chromosome across from? Its partner. Okay? It's homologous chromosome. If red is from mom, then blue is from what? Dad. And they line up across from their partner. Always. So instead of us having 2 rows of 3 pairs, we would have 2 rows of how many pairs? How many pairs? 23, right? Instead of having 2 rows of 3, we would have 2 rows of 23. Now here's the cool thing. Every time they line up, Dad's chromosome could line up over here, or it could line up over here. Same with mom's. So if you have 2 sets of 23, how many different ways could they line up across from each other? Yeah, well, it's about that. 2 to the 23rd power, which is somewhere around 8 million different ways. So they could line up across from their partner in 8 million different random ways. That's called independent assortment. And that's another way that what gets created? Variation. Because what's going to happen to these two sets, the set on the left and the set on the right, what's eventually going to happen? They're going to pull apart and they're going to end up in different cells, right? So that essentially means just because of this, we can make that many different sperm or eggs apiece. And that's not even thinking about crossing over. All right, that's metaphase 1. Anaphase 1 is sort of what anaphase always is. Anaphase is about what? Separation. Now in this case, the egg-shaped chromosomes, they don't break. What gets separated in anaphase 1? This chromosome is getting pulled that way. This one is getting pulled that way. Homologous chromosomes get separated during anaphase 1. The X's don't break. The X's are still together. The partners get separated from each other. And finally, what comes after anaphase 1? No. Telophase. It's PMAT, right? Prophase, metaphase, anaphase, and then telophase. And the telophase is always the same, pretty much, regardless of the kind of cell division. It's kind of the opposite of what stage? Opposite of prophase. So what happens to the DNA? It unwinds. What happens to the nuclear envelope, the nuclear membrane? It shows back up. It reforms. What happens to the spindles? They go away. And we end telophase 1 with one cell with two what? What does this one cell have? Has two what? What's this and this? Nuclei. It has two nuclei. Two nuclei that are the same or two nuclei that are different? Two nuclei that are different. Just like always, what comes after telophase? Cytokinesis. So then the cell divides. We end up with two cells. These two cells now are N. And in us, it wouldn't say N equals 2. In us, it would say N equals what? What's your N number? What's your haploid number? 23. So in us, after meiosis 1 is over, we would have two cells. Each one would have 23 chromosomes in them. And they're going to be the bivalent, the X-shaped chromosomes. And that's sort of where meiosis 1 is. What usually happens after cytokinesis in mitosis?
It starts over again.
What starts over?
Okay, cell cycle.
Yeah, what's the beginning of the cell cycle?
Okay, two.
Interphase, right? Interphase. But here, interphase is really short. And what doesn't happen during this interphase that usually happens in interphase? The DNA does not get copied, because if it did, that would sort of put you back at what? It puts you back at how many chromosomes? If you had 23 and you did S phase, you'd be back at what? You'd be back at 46. That's not where we want to go. So during this interphase, or what they're called interkinesis here, the DNA doesn't get copied. It goes straight into part 2 of meiosis. Now here's the deal with part 2. Meiosis 2 is pretty much just like mitosis. Nothing weird happens. We have two cells. Two cells we made in meiosis 1. Prophase 2, the normal prophase stuff happens. And again, what happens to the nuclear envelope in prophase? It goes away. What happens to the spindles? They form. What happens to the chromatin?
It winds up.
Winds up and makes chromosomes. In the little nucleolus, what's going to happen to it? It goes away. There's no crossing over. There's none of that stuff happening here. This is just like mitosis. And then we jump down to metaphase 2. What always happens in metaphase?
The chromosomes line up.
They line up in the middle. And notice in both of the cells, they line up in the middle. But this time they're lined up how? Single file. Because what's not there anymore? What have we already gotten rid of?
Pairs.
The pairs. There's not any pairs. So in you, there wouldn't be a line of 2, there would be a line of what? 23. You'd have 23 in a line here, and you'd have 23 in a line there. And remember, is this set of 23 on the right the same as that set of 23 on the left? No. What happens after metaphase? Anaphase. Remember, anaphase is all about what? Separating. In this case, when the spindle's shortened and tightened, the bivalent chromosomes, those X-shaped things, get pulled apart. So this set of DNA goes this way. This set goes this way. But it's also happening in the other cell. This set's going that way. This set is going in that way. And remember, it wouldn't just be 2 in each of these circles. It'd be 23 here, 23 here, 23 here, and 23 here. That's if it was happening in you. Then we get telophase. The normal telophase stuff happens. Opposite again of what? What's telophase opposite of? Prophase. Nuclear envelopes reform. Spindles go away. Chromosome goes back to being chromatin. Nucleolus reforms. We end this telophase with 2 cells, each with 2 what? What's that and that? A nucleus. Okay? 2 cells with 2 nuclei. And that's followed up by cytokinesis, which divides each of those cells in half. So we end up with 4 cells. We started with 1 diploid cell. We end up with 4 what? Haploids. And remember, are those haploids just like each other? They are very different, because one of the main purposes of meiosis is to do what? Create genetic variation, to make different kinds of things. That's meiosis. Now we've got a couple other things we want to look at here, and then we'll watch another little video. Let's talk about this, gametogenesis. Weird sounding name. What's Genesis? Like the Genesis in the Bible. What does that word mean?
The beginning.
Nope. Most people say that. Every time I ask that question, I'm asking for a lot of times. Most people say the beginning. It's not what Genesis means. It means creation.
That's what I mean.
Creation. Okay? So it means creation. This word means creation of what? Creation of gametes. It's gametogenesis, the creation of gametes. Meiosis is the main part of that, right? Meiosis does most of the gamete creation. But it works a little bit differently in men than it does in women. In men, it's called spermatogenesis, which means creation of guess what? Sperm. In women, it's called oogenesis or oogenesis. Anytime in biology you see 2 O's together, focus for me. You see 2 O's side by side in biology. It means egg. Creation of eggs.
So like chicken eggs?
Eggs in general. Right now we're talking about people though. Alright, now we're going to look at a diagram of how this might work. Here's spermatogenesis. This is the way it works in a guy. These cells right here are what we've been calling germ cells. These would be in your testes. And these cells have 46 chromosomes inside them. And right now, after replication, they're going to be X-shaped chromosomes. Those go through meiosis part 1. And remember, meiosis part 1 takes 1 cell and divides it into 2. And we called it reductive division because what does it reduce? What does meiosis 1 reduce? The number of chromosomes. So these 2 cells are going to have how many chromosomes in them? 23. 23 here, 23 here. But they're still what shape? They're still X-shaped. They hadn't divided yet. Meiosis 2's job is to take these X's and do what to them? Divide those. So this goes through meiosis 2, and this one goes through meiosis 2. We end up with 4 cells. These still have 23 chromosomes each. But what are those 23 shaped like? They're half of what? Half of an X. They're a single chromatid. Those 4 cells grow up. They go through this process of maturation to become sperm. Sperm that are different from each other. One germ cell, one diploid germ cell, creates 4 haploid sperm cells that are different from each other. That's the way it works in a guy. Now, in a lady, it works a little bit different. All right, here's the deal. You ladies, your germ cells are called primary oocytes. They're diploid, 2N, and they have 46 X-shaped chromosomes after replication. All of your ladies' germ cells have already done replication. They've all also already gone through meiosis 1. Now I want you to notice something. When they go through meiosis 1, that one cell splits into 2. But it splits into one really big cell and one little bitty cell. Each one of these have 23 chromosomes in them, I'm sorry. But it's both still X-shaped. Any idea why it didn't divide down the middle like it did in a guy? Why did it divide into a big and a little? What's going to happen to that little cell? That little cell is going to die. It's basically just a trash bag for what? What's that little cell all about? What's its job? To get rid of what? What's meiosis trying to get rid of? Half of what? Half of the chromosomes. Okay? Those are just going to be like throwaway chromosomes. 23, get out of here. Okay? That little cell is called the polar body, and it will die. The big cell, it's going to go on and divide again. And that big cell, but it never does that in you ladies unless a sperm comes in and gets through the first couple of layers of that egg cell. So most of your eggs, ladies, most of your eggs never make it to this part. Most of them die. Okay? They die like this. But if a sperm gets through that outer coating, then that egg will divide again. And notice it divides again into a big cell and little bitty cells. This big cell now is going to have 23 chromosomes in it, but they're going to be single chromatid chromosomes. These are polar bodies. They die. Their job is to get rid of extra DNA. So you ladies from one germ cell really only make how many working egg cells? 1. So 1 to 1 for you ladies. For us guys, it's a 1 to what? 1 to 4. Why does that egg cell need to be big like that? Why does it divide big and little? Why is an egg so big?
Because it's going to be a big person.
They have to hold a lot of what inside them?
What?
No? They have to hold a lot of food. Think about this for a minute. Where in a woman's body does fertilization of the egg happen?
The uterus.
Oh.
Yeah.
Stomach? I don't know much about women, do you? It happens in these little tubes called the fallopian tubes. Okay? Little curvy, windy roads. Okay? The fallopian tubes are the connections between what two things?
The ovaries and the uterus.
Ovaries and the uterus. So the sperm have to swim through the uterus, up the fallopian tube, and they find the egg. Join me. We got a recording on here. Let's be quiet. So the egg and the sperm get together inside the fallopian tube. They make a zygote. Then nuclei fuse. That zygote now is diploid, and it's got 46 chromosomes in its nucleus. It's the beginning cell of a baby. But how long is it going to take that egg to get down the fallopian tube, into the uterus, and grow a placenta and an umbilical cord and all that kind of stuff? How long is that going to take? It's going to take weeks. What is that zygote feeding on between fertilization and when the placenta and the umbilical cord actually start working? It's feeding on whatever was in the egg to begin with. Which means the egg has to be big so it can store what? Food. And what do we call that stored food?
That
Oh.
Yolk. You ladies have yolk, just like an egg has yolk.
You're like
It's stored pretty good. Wait up. Yeah, give it to me. So here's the deal. The egg needs to be big so it can store food. The egg's jobs are to deliver 23 chromosomes, but also to store food for that baby and to supply all the organelles the baby will have. The sperm's job is just to deliver what? 23 chromosomes and it's done. Okay? So it's small. Its job is just a chromosome delivery. That's it. Let's go back. Aaliyah wanted to see that slide. One more back. You really don't. If you know the two pictures, you really don't need that one.
Oh, okay.
That's just describing the pictures. All right. So let me stop the recording. All right.